Hello, grade 8 student. In this presentation, we're going to talk about the different food tests and their indication, about the different digestive enzymes, and the proper conditions for the normal functioning of enzymes. So let's start with chapter 1, activity 1, our food. So what do we eat and what happened to the food that we eat it? First of all, let's start with digestion. Digestion is the transformation of complex food molecules into simplest one to be used by the body cells. Which system is responsible for digestion? Sure, it's the digestive system that is divided into digestive tube or digestive glands. Digestive tube, organs through which food passes through. Digestive glands, organs through which food doesn't pass through. Digestive system includes organs of digestive tube and organs of the digestive glands. Digestive tube organ they are mouth, esophagus, stomach, small intestine, large intestine, and finally the anus. While in the digestive glands we have the salivary glands, gastric glands, pancreas, liver, and the intestinal glands. So, what's the function of digestive glands? Digestive glands they secrete digestive juices into their site of action. Let's move to this table talking about the different digestive glands, their juices, and their site of action. So salivary glands, it secrete a juice is called saliva, where inside the mouth, where the medium is neutral. Gastric gland secrete a gastric juice in the stomach, where the medium is acidic. Pancreas secretes pancreatic juice. Liver secretes bile. Intestinal glands they secrete intestinal juice in the small intestine where the medium is basic. So let's here talk about neutral, acidic, and basic. Neutral meaning that the medium is neutral pH is equal to 7. Basic pH greater than 7. Acidic less than 7. Now let's move to the food, organic and inorganic branches. Organic, it contains carbohydrate, proteins, lipids, and vitamins. Inorganic, it contains water and mineral salts. So let's move to the category of food, complex, intermediate, and simplest form. Carbohydrates and proteins and lipids. First, let's talk about digestion and its complex definition. So digestion is the set of chemical and mechanical transformation of food molecules into simple nutrients to be used by the body cells to produce energy needed for cellular activity. Carbohydrates, the category of organic food. Complex form, it's polysaccharides. It's a complex chain of simple sugars. It contains starch, cellulose in plants, glycogen in animals. Intermediate form, disaccharide, it's made up of two simple sugars bonded together, maltose, glucose and glucose, lactose, glucose and galactose, and sucrose, it's glucose and fructose. Simple form, it's monosaccharide, simplest sugars made up of one unit only, glucose, fructose, galactose. All of them they are reducing sugars. Moving to the second category, proteins. Proteins, the complex form is protein, complex chain of more than 52 amino acid, polypeptides, chains that contain less than 52 amino acid, and the simplest form is amino acid, it's made up of only one unit. The another category is lipids. It's very energetic. It has the same quantity of lipids as has double the amount of energy supply but the same amount of carbohydrates. It has only complex and simplest complex form lipids, simplest form fatty acids and glycerol. How to identify the presence of certain categories of food? Food tests, aim, steps, positive, negative, analysis of the positive, and indication. Water test, it aims to identify the presence of water. Steps of the test, heat the food in a test tube. Positive result, presence of water droplets. Negative, no water droplets. Analysis, after heating the food, water droplets are formed on the walls of the test tube. This indicates the presence of water in the food. Salt test is done to identify the presence of sodium chloride. Add distilled water to the food in a test tube. Shake it well. Add silver nitrate AgNO3 to the tube. Positive result, white precipitate appears, silver chloride crystals. Negative, no white precipitate appears. Analysis of the positive. After adding distilled water and silver nitrate solution to the food, white precipitate appears. This indicates the presence of salt in the food. Fanning test is done to identify the presence of reducing sugar, glucose, fructose, galactose, maltose, lactose. Add equal number of drops of fanning A and fanning B. Heat the contents of the tube. Positive result, brick red precipitate appears. Negative, solution remains blue in color. So the analysis of the positive result, after adding equal number of drops of fanning A and fanning B and heating, or simply you can say after applying fanning test, a brick red precipitate appears. This indicates the presence of reducing sugar. Iodine test is done to identify the presence of starch. Add a few drops of iodine solution. A dark blue color appears. No dark blue, which is a negative result. After adding iodine to the food, dark blue color appears. This indicates the presence of starch. Biuret test is done to identify the presence of proteins and or polypeptides. Add equal numbers of drops of NaOH and CuSO4. Violet color appears. The color remains blue in color. This is a negative result, while the violet is a positive. After adding equal drops of NaOH and CuSO4, or after applying, you can say Biuret test, a violet color appears. This indicates the presence of proteins and or polypeptides in the food. Coagulation test, done to identify the presence of proteins. Heat the food till boiling. A positive result, the food coagulates. Negative result, no coagulation. After heating till boiling, the food coagulates. What does this indicate? This indicates the presence of proteins in the food. Translucent spot test is done to identify the presence of lipids. Rub a piece of food against a paper. Observe it under sunlight. Translucent spot is formed. This is a positive result. And no translucent spot is formed. This is a negative result. After rubbing the food against the paper and observing it under sunlight, a translucent spot is formed. This is the analysis. This indicates the presence of lipids in the food. Sodium test is done to identify the presence of lipids. Add sodium test solution. Pink color appears. This is a positive result, and negative, no pink color appear. So after applying the sodium test, pink color appears. This indicates the presence of lipids in the food. Interpret the results of document D and indicate the components of each of the tested food. So after applying iodine test to tested food, dark blue color appears with banana and potatoes, but not with fish and grapes. What does this indicate? This indicates the presence of starch in potatoes and banana, but not in grapes and or fish. After applying fanning test to tested food, brick red precipitate appears with the grapes, but not with potatoes, banana, or fish. This indicates the presence of reducing sugars in grapes only. After applying Biuret test to tested food, slight violet color appears with the grapes and strong violet color appears with fish, yet no violet with banana and potatoes. This indicates the presence of proteins and or polypeptides in fish and grapes, but not in potatoes and banana. So we can indicate that grapes contain reducing sugars and proteins, banana and potatoes contain starch, and fish contain proteins. Before moving into activity 2, we have to keep in mind that digestive juices, except bile, contain enzymes. These enzymes, they are scissors. They cut, they hydrolyze, they digest, they transform, they break down food. These catalysts they remain unchanged at the end of the reaction, and they facilitate or they speed up the reaction, they make it faster. Table of digestion and organ. This is a very important table. Mouth contains a salivary gland that secreted juice called saliva. This saliva contains an enzyme called salivary amylase that works on starch to give maltose. Medium is a neutral, pH equals 7 and temperature 37 degrees Celsius. Stomach contains a gland called gastric gland. Gastric gland contains a juice called gastric juice. This juice contains an enzyme called pepsin. Pepsin works on a protein to give peptide 3P, as I said before. 3P, pepsin, protein, peptide, medium, acidic, temperature is 37 degrees Celsius. Small intestine contains pancreas that secrete juice called pancreatic juice. So we have 4 enzymes in the pancreas. Peptidase, as its name indicates, it works on peptides to give amino acid, medium basic, temperature 37. Also pancreas secretes pancreatic juice containing enzyme trypsin that works on a protein to give amino acid, medium basic, temperature 37 degrees Celsius. So trypsin and pepsin, both of them works on protein, but trypsin in basic, pepsin in acidic. Pepsin digests protein into peptides, while trypsin digests protein into amino acid. Pancreas also secretes pancreatic juice that contains an enzyme amylase that work on starch to give maltose. Medium is basic. Difference between amylase, pancreatic amylase, and the salivary amylase. The salivary amylase works in a neutral medium, while the pancreatic amylase works in a basic medium. Also, pancreas secretes pancreatic juice that contains an enzyme called lipase that works on lipids to give fatty acids and glycerol. Medium basic, temperature 37. Liver secretes an juice called bile. Bile, as we said before, it's the only juice that contains no enzyme. So bile works in the emulsification of lipids. It removes the particles of lipids aside. It distributes the particles of lipids to be digested by the enzyme lipase. Here we have intestinal gland, also contain intestinal juice that contains enzyme erepsin that works on peptides to give amino acid, medium basic, temperature 37. Also, intestinal juice contains an enzyme called sucrase. Sucrase, as its name indicates, sucrase works on sucrose to give glucose and fructose, medium basic, temperature 37. Also, maltase works on maltose to give glucose and glucose, medium basic, temperature 37. Also, lactase works on lactose to give glucose and galactose, medium is basic, temperature 37. Intestinal juice contains lipase, works on lipids to give fatty acids and glycerol, medium basic, temperature 37 degrees Celsius. As we see here, all of the enzymes, they work in a basic medium except for pepsin and salivary amylase. Salivary amylase, it works in a neutral medium, and pepsin works in an acidic medium. Chapter 2, Chemical Transformation of Food. This experiment explains what we have to know about this activity. We have 2 tubes A and B are placed in a water bath, temperature 37 degrees Celsius. Both of them contain cooked starch. Tube A contains a small amount of fresh saliva, while tube B doesn't contain. So what's the role of tube B? It's called a control tube, used to compare and explain the different experimental results. Second, what is the variable factor? Saliva. What is the role or the aim of the experiment? Is to study the effect of saliva on the digestion of starch. What is the problem? It's the same as the aim, but should be in the form of question. So what is the effect of saliva on the digestion of starch? Moving to the table of conditions and interpretation for this experiment. So after 15 minutes at time 0, we did iodine test. We have dark blue, so both of them contains starch. After 15 minutes with iodine test, tube B remains dark blue, meaning that it still contains starch, while tube A, the dark blue color becomes brownish or orange in color. In failing test, both tubes at time equals 0 gives a negative result, dark blue color, while after 15, only tube A gives red precipitate, meaning that digestion of starch occurs only in tube A and not in tube B. So what is the role of the tube B? It's a control tube, substrate, starch, variable, saliva. Aim to study the effect of saliva. Pose a problem. What is the effect of saliva? Describe document A on page 18 using the passive voice for sure. Compare the content, construct table, interpret, deduce the role of saliva, indicate the trigger. So tube B, as I said, is control tube. It's used to compare and explain the result of the experiment. Number two, what is the substrate? Is the starch. Variable factor, saliva. Why? Since it's present in tube A and it's absent in tube B. Aim to identify the effect of saliva on cooked starch. 5. What is the effect of saliva on cooked starch? 6. Two test tubes A and B are brought. In both tubes, cooked starch is added, but to tube A, only a small amount of fresh saliva is added. Both tubes are placed in a water bath at temperature 37 degrees Celsius. Content: both tubes contain cooked starch. Tube A contains fresh saliva, tube B doesn't. Table of condition: you have to put all the conditions: substrate, starch, saliva, water bath, temperature, time. Legend: plus means presence, minus means absence, and you have to put a title for the table. Interpret: it's analyze plus an indication. So at time 0, after applying iodine test, both tubes A with saliva and B without saliva show dark blue color. This indicates the presence of starch in both tubes. Yet, with Fehling test, both show blue color. This indicates the absence of reducing sugar. After 15 minutes, tube A with saliva show orange color with iodine, while tube B without saliva show dark blue. Moreover, tube A shows brick red precipitate with Fehling test, while tube B shows a blue color. This indicates that the presence of saliva starch in tube A is transformed into reducing sugar, but in tube B without saliva it remains unchanged, it remains the same. So what can we deduce about the role of saliva? Saliva is responsible for the digestion of cooked starch and to reducing sugar. And the role of the last sugar of the final product, it's maltose. Thank you everyone. Bye bye.